because of the Women's Week theme, I was asked to talk a little bit about sort of my career as a woman and then some of the science. So the first part is about women in science really and my experiences, and then but the most of it will focus on some science. And I know this is a mathematical institute, but I'm a biologist at heart, and so to be reasonably biological, but please jump up and wave if I'm talking about things you don't understand or that you're not sure of. So just to give a bit of history about women in science. So when the universities first started in the 11th century, women were excluded from science and from universities. So Laura Bassi was an Italian scientist who was the first woman to earn a university chair in a scientific field at a university. This was in physics. But then despite this, there were a lot of advances made by women in science in the 18th century. And in the 19th century, they were still excluded from universities, but were allowed to join some learned societies. But then, of course, most people know one of the most famous women is Marie Curie in 1903, who won a Nobel Prize for physics. And then between 1901 and 2010, in fact, 40 women have been awarded a Nobel Prize, 16 of these in science. So things have progressed. There's three famous women in science. So Marie Curie, she got the Nobel Prize for discovering polonium and radium. So this has basically led to X-ray technology. Rosalind Franklin, she worked on X-ray diffraction images of DNA. She actually was one of the key people, even though she wasn't recognized at the time, for discovering the DNA double helix, which is extremely important to genomics and genetics now. And then Gertrude Elliot, she did a lot of work in drug discovery, and she actually, her research led to the development of the AIDS drug, AZT. These are just three out of obviously a very large pot of famous women in science. So if you look at some of the research that's been done, so this is from the Association for Women in Science, and this is on a particular set of biotech companies in the US. And actually, the research shows that there is an important impact of women in science. So, for example, they showed that companies with more than 15% of the top management positions occupied by women had a 52% greater returns on equity than those with less than 10%. Companies with at least one woman director perform 17 to 26% better in their stock prices. And I know the men will not enjoy this one, but several studies have shown that regardless of individual IQ, teams that include women have a higher levels of group intelligence than teams comprised solely of men. And this is their research, not from me. So research has shown this to be true.
Sorry, but technically those first 2 points show nothing. They show
I know.
Association but not causality.
No, I know. There's a whole bunch of research, and they're just trying to... Yes, nevertheless. Okay, but despite this, despite some research that's been done, there's still a major problem. So some recent articles in some reasonably good journals show that women, although highly qualified, they still don't get the good science jobs. And Nature is saying that even though there is this increased sort of push towards women in science, they get lower salaries, they're not getting the positions they want to, and they're just not getting the opportunities of similar qualified men. So it's still a problem. I think it's for the camera.
Can we hit the other one?
Yeah. Okay. Is that better? Okay, so some of the challenges for women in science, and this is things that I've come across with our network and in our environment, is social and cultural challenges. So in our Pan-African Bioinformatics network, there are several women from Muslim countries that have some cultural and religious challenges. Obviously, women have to take time out to have children if that's their choice. And there are family responsibilities. Children get sick. People have to deal with them. And then competing in the workplace. You've always got to compete with men. And internationalization of science, this is something that I find now, is that I have a lot of collaborations with all different countries. And so I have conference calls because I collaborate with the US, with Canada and Australia. I have conference calls at 7 p.m., which is the worst time for family time. And many conference calls starting at 10, 11 p.m. So it is actually not conducive to family life. So my career, it's never really, I have to say, it hasn't really affected me. I think you've heard already from the introduction where I was and where I am now. So now at UCT, I've managed to build up a group of about 30 people there. And during my time, I had some good mentors at EBI. The director of the EBI was a woman, and she was really good mentor. But frankly, I just took opportunities that arose. I just didn't let it affect me that they were, I was possibly competing with men. I just treated it as though I was competing with anybody else. And that actually worked. In our division now, computational biology division, is reasonably evenly split between males and females. And this was not the case when I first started. So when I first started, there were probably 90% males and much fewer females. So we are seeing an increase in females in this field. But in our Pan-African Bioinformatics Network, the split is a bit different. So there's 30% female, 70% male. But I have to say that of the young upcoming academics, the most promising are the young females. So that's enough about women in science. So moving on then to some of my research about the science of women. So in this Pan-African Bioinformatics Network, basically what we're trying to do is build capacity for genomics research. And genomics is quite challenging because it's expensive to generate the data. The data is very big and complex, so it's a big data problem. And we have ethical issues in human genomics research because the data is very sensitive. But we cover kind of a wide variety of genomics research. And what we're trying to do in this H3A Bionet is actually to build capacity for this. And we're actually training a lot of both women and men in Africa to be able to analyze our own big data on the continent rather than send it overseas for collaborators. So my research is focused, the genomics research is focused on two different parts. The human genetics, which I actually gave the last public lecture I gave here was on that a few years ago. So now I'm going to talk about a new field that we have moved into, and that's the field of microbiomes and metagenomes. So metagenomics is basically the study of all those little bugs, all the bacteria, all the viruses, all the things you can't see on something. So basically, you take an environmental sample and you sequence it. This means you work out what the DNA is in there, and you try and work out what organisms are in that sample. So there have been many, many metagenomic studies to date. So the most famous ones were done on the ocean samples. Somebody went out on a yacht, took samples, and sequenced what was in the ocean. But since then, there have been many different examples. There's a high-impact journal paper on windshield splatter. Somebody literally scraped the windshield of all the bugs and sequenced those to see what you get on the windshield in different parts of the country. And this was in the US. People are doing microbiomes on metagenomic studies on subways, on the deep mines, in animals, in humans. So people are trying to work out what is around us. And the reason they do this is because you can now sequence these directly. You just need to take that mush of what you find and sequence it. Previously, you had to try and culture it, and you could only discover what you could grow. Now, even if it doesn't grow in the lab, you can still discover it because you can sequence it directly and see what it actually is. So you could either do this.
Do you mean genome sequencing?
Genome sequencing. So you can either do this by full genome sequencing, that's shotgun sequencing, or you can do it by targeted sequencing. So this targeted sequencing is basically every organism in every bacterial genome has the gene called 16S ribosomal RNA. It's just a gene out of a few thousand in every single bacterial genome. Now, this gene happens to be very conserved across all of these bacteria in most of its sequence. But there are several variable regions in there which are highly predictive of what type of bacteria it is. So if you take a whole bunch of bacteria, you mix them together and you sequence them, just this region of this particular gene, you can then work out exactly what species of bacteria are in there, even if you couldn't even grow them up, because of our knowledge of the variation in the single, otherwise highly conserved gene, which happens to be in all these bacteria. So a lot of metagenomics uses 16S rRNA sequencing to try and distinguish. The idea is to distinguish what is in this mush.
Is it also in plant matter?
Everything. Bacteria are everywhere. And then what you do basically is you take this, you get the DNA out, there's a technology to pull out just that region of that gene, sequence it, and then the analysis part is quite computationally intensive, it's quite complex, you have millions and millions of reads that you've now got to piece together and work out what they are. So it's basically like putting them into bins to say, okay, is this part from this bug, or from this bug? And then you basically get what you end up with at the end is these operational taxonomic units, so OTU, and I'm going to use that term a few times. basically that's the list, in simplified terms, it's the list of bacterial species that you are finding in your sample. Okay.
Can I ask you what rate, well, I suppose one should ask about a particular study, but do you find, what's the proportion of previously unknown, taxonomically unknown species?
So again, that's still quite difficult to find because in the pipeline, what you've got to do is you have to actually map these short reads to something that exists in the database. So you have a database of reference genomes.
So if you find something that doesn't appear in the database?
You can do what's called de novo assembly, and that'll basically now try to assemble based on overlaps of the reads anything that wasn't mapping to a reference, and that'll give you an indication of what's novel. But people tend to actually want to know, in many of these cases, they are reference databases because so many of these studies have been done now. They've built up such big reference databases of many organisms which you can't culture, but now they've seen them somewhere in the world in some metagenomic sample.
The rate of novel material is decreasing as the number of studies
Yes.
increases.
Yes. Yeah. So this is basically the analysis pipeline. So you take your sample, you get your DNA, you sequence it, then you run it through this pipeline to say, based on my variation I'm seeing in this gene, these are the sets of organisms that are present, or the OTUs, and then you're normally comparing that to something. So you want to now compare two different metagenomic samples. What do you find in the deep mines versus Antarctica or something? So there's always some downstream statistics. So one aspect, one kind of specific type of metagenomics is microbiomes. And this is, so in this Nature article, it's basically called Our Other Genome. And the reason for this is that by weight, you are more human than bacteria because your cells are bigger, but by numbers, it's not even close. So we are made up of more bacteria than we are human cells. And bacteria are everywhere. So if you don't like bacteria, you're on the wrong planet. And if you look at, so the microbiome is basically the study of these microorganisms in a body. So either in the human microbiome, people are now studying animal microbiomes, plant microbiomes, because they have a huge impact on agriculture. So the number of microbes in the human body outnumber our own cells by 10 to 1, basically. And what they're finding is that the changes in this microbiome are actually having a huge impact on our health and many other things around us. So restoring the microbiome is a way to cure many diseases, in fact. So because of this importance of the microbiome, the NIH funded a very big human microbiome project, and this is actually hundreds of research groups around the US to sequence various parts of the human microbiome. So they looked at it, they started off with 5 major regions, which I'll show you on the next slide. By 2012, they had over 2 terabytes of data, and this is only 60 days data, so it's a very small data set compared to normal sequencing data sets. And they had 300 subjects from 15 body sites, so they had extended it. So the first microbiome studies were done on the nasal microbiome, the oral microbiome, skin, the gut, and then urogenital microbiomes. So this is the, just an overview, obviously you can't read this, an analysis of those different body parts, and this is the phylogenetic tree. So when you have a set of organisms, you can try to work out how related they are to each other. And this is basically telling you what microbes are the most common in different parts of your body.
Sorry, can you, just from the previous one, can you explain that graph just a little bit, or just give us
Okay.
a few of the salient details there?
So these are the different body types. So that's vaginal, there is gut. And in those, they are, these are all the bacteria that you find in the microbiomes. Okay, and this is, so the phylogenetic tree shows you how related they are. So if you saw a normal phylogenetic tree of human, of apes to human, which basically it's just a tree demonstrating the relatedness, evolutionary relatedness of different organisms. So this is just showing how these organisms are related to each other, and the ones that are grouped by the ones that are coming from specific parts of the body. And they are actually quite, so if you look at the next slide, if you do a PCA analysis, they're quite different sets of organisms in different parts of our body. So you see principal component analysis. So basically it's a way of splitting up data to show where the variation is. So you can see that there's a cluster of microbiome data for the guts, urogenital, skin, nasal mixes a bit with skin, and the oral microbiome is quite separate. So you can see that all your body, your different parts of your body is colonized by different kinds of microbes. So everybody varies in their microbiome. And this variation, I mean, what they notice, and it's really difficult statistical problem because you try to see what is a normal microbiome, and actually the individual variation is so huge that it's difficult to see. But with more samples, they are starting to find an approximately normal microbiome. But it changes with age, puberty or pregnancy, diets, medical conditions, what drugs you're taking, environmental exposures. So there's a number of things that affect the microbiome. And the way it varies is in what kind of microbes are present, the relative amounts of each of these particular species, and then the genes from these. So what genes are actually made, what proteins are made by the set of organisms in your microbiome. So diet affects the microbiome, and the microbiome affects diet. So what many people don't know is that a lot of the things you eat are not actually degraded by you. They are actually degraded by your microbiome. And so this is the human gut, and these are all the plant, these are the different materials that you ingest. It's plant material, there's animal material, and actually a lot of the degradation and the metabolism of this is actually being done by your microbiome and not by you. So there are many studies on the effect of diet. So this anthropologist has started looking at whether the hunter-gatherers have a key, whether their microbiome is the key to a healthy lifestyle because of their diets. And actually their diets will change the microbiome, and that will then affect what they can then further metabolize. So it's a very much a strong relationship between your microbiome and your diet. But the most important thing and why we study microbiomes is because there's a link between microbiomes and diseases. So microbes are these obviously you have them, so your microbes are your friends, but they are also pathogens, and also even the ones that do exist in there, if there's a dysbiosis, so there's a change in the proportion, that could trigger a disease. And so what happens when you're born is you get your microbiome develops in your gut, and your immune system starts to get trained of what is the good stuff and what is the bad stuff. So it can recognize a pathogen coming in as opposed to what is already there. And so it distinguishes self from non-self, and where self is now what your own microbiome is. And then the microbiome also has an impact on the immune system, so it secretes things that actually impact how you respond immunologically to diseases and to vaccines. So some examples of links between microbiome and disease. So the skin microbiome affects psoriasis, acne, dermatitis. The gut microbiome has a whole range of different diseases related, including irritable bowel syndrome, colon cancer, gastritis, diabetes, obesity. Very strong links have been found between these diseases and the gut microbiome. And then sexually transmitted diseases, bacterial vaginosis, all in the urogenital microbiome. These are just a few examples. I'll show you a few more. Some of so these are just a few of the others. So heart failure, metabolic syndromes, and then diabetes is a big area of study at the moment. So just to go on then to some of the research that we're doing. So this is just for the women thing. This is all done by a group of women. So Katie Leonard's a postdoc in my group, and these are collaborators at UCT. And so they're looking at, this is the science of women, the female genital tract and susceptibility, HIV susceptibility in young South African women. So basically, although HIV incidence or transmission is decreasing worldwide, actually in young South African women it's not decreasing, and the rates of transmission is actually about 8 times higher than in males of the same age. And they've said it's not a behavioral thing because they've looked at the data. So there are hypotheses that it might be related to sexually transmitted infections, changes in hormone, or the microbiome that is actually changing this risk of HIV infection. And this is just a paper that has been submitted to show some connection between previous inflammation and HIV susceptibility from the same collaborators. So the healthy female genital tract is occupied by lactobacillus species. There's low diversity and there's low pH. If you change that, you tend to get bacterial vaginosis. I've just got a BV from now on. And it's associated with high levels of inflammation, and it causes increased susceptibility to HIV and other infections. This is just a cartoon of the region. This is the normal healthy genital tract, and this is where there's dysbiosis in the microbiome, and it causes either from changes in the number of CD4 cells or the immune cells, or this is the hypothesis that it's either that or actually just changing the function of this region and the related immune factors that are causing increased susceptibility to HIV. So this study took about 300 women, young girls of about 16 to 22. 161 of them had their microbiome sequenced, and they had detailed information about inflammation, so the cytokine measurements, and then metadata, so their behavioral data, contraceptives, BB status, any other infections they've had. And you can see that so there is a test for BV, and this is called the Nugent score. It's actually done by microscopy, where they work out whether you have bacterial vaginosis or not. And in that case, it's not very easy so they give you a score called the Nugent score. And then, so negative would be extremely low or zero BV score. Positive will be a very high score, and then there's intermediate, which is sort of somewhere in between. And you can see that the microbiome, so this is basically these are different classes of bacteria. So this is showing which different classes of bacteria are in this microbiome. And you can see that the negative ones are, as we said, what the healthy one should be, mostly lactobacillus, not much diversity. Intermediate is something between the two, and BV, you have this massive range of different bacteria there all of a sudden.
Is Shuttleworthy anything to do with Mark Shuttleworthy?
No.
Okay, thank you.
Not that I know of. And again, if you now separate this out to look at the variation, you can see that it separates reasonably, although there's a bit of a mix. So the red is BV, the blue is negative, and then the green is intermediate. So the main question is we asked was, could we actually use the microbiome to classify as to whether you've got BV or not, and is it actually better classified than the original lab diagnosis? And so many other labs have worked on this, and they've classified it to say that they've got typical classifiers. And they've seen that healthy ones, you normally have Lactobacillus crispatus, it's a specific kind of lactobacillus. Intermediate is a different kind of lactobacillus, and then a BV is completely mixed. And so we sought out to see whether we could use this classification. And in their classification, these are the different groups. That's the negative, the intermediate, and the positive. And here, these are the main bacteria that you're seeing in there. And they were able to distinguish these and to correlate these with immune factors. So in our data, again, we used that classification. And you can see from, these are the red is the positive, white is negative, and gray is intermediate. And it kind of classifies it so that all the positives are on one side. It's not 100% accurate, though. So we looked then to see, well, you can do two things. You can either look at the microbiome, so how many different species are there and in what proportion. That's what the data we've been looking at to date. But you can also now see what genes are in there, so what functions are in there. So if you take all the bacteria that are in there, you know what genes they have, because we know we've sequenced the genome. So we know what every gene for every bacteria is. Can we look at what these genes are doing? So in other words, we're looking at the functions of the microbiome. And actually, if you look at, this is from the Human Microbiome Project, if you look at different samples, this is the variation in the microbiome across different samples. You see they're quite different, these blocks. So where the different color is a different class of bacteria. So these are very different to each other. But if you look at the functions of those same bacteria in the different samples, the functions are a lot more coherent. So the overall functions in the microbiomes are very similar, even though the species themselves are changing. So we decided to look at the functions of the microbiomes to see if they are classifiers or not. And this is just how you do it. And if you look at that, there was, again, a separation of the data. So separated by Nugent score. So this is the BV positive, very high Nugent score, intermediates, and then the darker ones are the BV negative. So they do separate out, and this is now not by the amount of and presence of different organisms. This is by the function of those organisms that you're seeing. And this is just to show that there was significant difference between those different functions.
Sorry, could you give an example of a function of an organism?
Yep. So this is now, we looked at the actual specific functions of the organism. So an organism will do many things. It has to make DNA, it has to make proteins, it has to metabolize things, it metabolizes carbohydrates, it has to move. So actually one of the major functions that came out as a classifier between these groups of intermediate versus very high Nugent scores was actually flagella assembly and bacterial motility. So movement of bacteria. This is a function of that came out to be significantly different between the two. But a function can be anything that the bacteria does, anything that we would do, which is move, like respiration, so breathing, metabolize things, and make all the basic things for life.
So were the BV positive group, were they more likely to have immobile or mobile?
So in this case, the flagella was more abundant in A, which was moderate.
Okay.
So it's less abundant, so they're less mobile in the BV positive, the stronger BV positives.
Okay.
So you can see that you can look at this based on the actual composition as well as the function, and in this case, this is actually seems to be a slightly better classifier for the different types of diagnosis. So we then looked at the relationship between this and immunology. So when you looked at the intermediate and the versus the very strong Nugent score, so very positive BV, there was no difference in the HIV target cells. So it wasn't actually the target cells that were being affected by the microbiome, but there was a difference in the cytokines. So this, basically immune cells, they need to go to the site and they then make specific molecules to cause an immune response, okay? There are different kinds of cytokines. There's inflammatory ones, and the inflammatory ones, so if you hurt yourself or you have a mosquito bite, you see it goes red. That is because the inflammatory cytokines come to the site and they start working on whatever's been infecting you or whatever the problem is. And it's these inflammatory ones which are actually have been shown to be causing HIV susceptibility. And it turns out that of the different cytokines measured, 15 cytokines were significantly increased in the high BV, so the very positive ones versus the intermediate ones. And these are the inflammatory ones that are in there. So basically, anyone who's who's strongly positive for BV has a lot of these, has this increased inflammation because of these cytokines that are coming out, which is probably what's causing the susceptibility to HIV. This is just to show it in, this is a correlation matrix, so you can see that these ones are, the ones in purple are the ones that were shown to be increased. And you can see that if you now look for which particular bacteria are correlated with the increase, so this is decreased in group C versus A, and these are increased in group C versus A. There's a very specific set of bacteria, so not only is the overall composition important, but the actual what bacteria are in there are actually influencing the immune system. So to conclude that study, basically these adolescents fell into three categories of negative, intermediate, and positive. The composition was very distinct, and the microbiome was very good at classifying this laboratory diagnosis. And although there was no difference in HIV target cell activation, there was a difference in the cytokines that were made. So it's probably the hypothesis is that the altered microbiome function changes this the barrier function that was at the where the cells are in the genital tract, and this basically facilitates HIV transmission. It basically weakens the cells so that the HIV can come in and infect. And then just briefly, a second study we did. So this was also looking at microbiomes, but this time we were looking at gut microbiomes of infants. And these are infants who were either HIV exposed or unexposed. They were all HIV negative, but they were exposed because their mothers were HIV positive, or they were unexposed because their mothers were HIV negative. And we looked at the impact of their microbiome, whether their microbiomes differed, and whether there was an impact on vaccine response. So there are several things known about the gut microbiome. So it plays an important role in immune system development. Feeding affects the microbiome. The maternal microbiome influences the neonatal microbiome. So particularly when there is natural birth, the skin cells get onto the baby, they tend to ingest it, and there is an impact. So they tend to start off with a bit of the mother's microbiome, which is very different to C-section births. And then HIV-exposed infants receive CTM. It's a prophylaxis against different bacteria. And HIV-exposed uninfected, so remember now from now on that HEU is exposed but uninfected versus HU, which is unexposed. So they're all uninfected, but there's exposed versus unexposed. And they've been shown to have a different vaccine response. So basically, a child who's been born to an HIV-positive mother has when you give them the BCG vaccine, which is for TB, they'll have a very different response to the vaccine compared to a child who's been born to an HIV-negative mother. So, as I said, we sought out here to study the gut microbiome, see if it was different between these two types of infants, and to see what the response is, how this related to vaccine response. So this was a pilot study done in Khayelitsha with 500 HIV-exposed, uninfected, 150 unexposed, and this was breastfed, unexposed breastfed. And then there was also a group of formula-fed, because we also at the same time wanted to look at the difference in the microbiome between formula-fed and breastfed to see, because it's well known that people have said that breastfed babies actually have a stronger immune system, but actually we didn't find that. And all the babies were HIV negative. So we went through the usual process. In this case, you take stool samples, so you literally have a lab full of poo, and somebody's got to scrape babies' nappies, and somebody's got to do this in the lab. And they isolate the DNA, and they sequence it with the 16S rRNA gene again, and then they do this OTU analysis. So they're trying to find what taxonomic groups or what bacteria are in these different samples. So again, if you look at this on how it splits out in terms of variation, you'll see that the triangles are formula-fed and the circles are breastfed, and you can see that they're actually quite mixed. So in fact, there was no correlation at all between the microbiome and whether it was formula-fed or breastfed. So we were not expecting that. We were assuming that formula-fed babies will have quite a different microbiome, but it turns out that after a few weeks it settles out and it's not affected at all.
But sorry, just on that, is it possible that the breastfeeding could have effects other than the microbiome on the child, which would themselves lead via another pathway to improved immune response? Yes,
so it would directly, because the mother's milk will have antibodies from the mother. So that's not going via the microbiome. It's actual direct transmission of antibodies, so immune factors.
So this can't be taken as proof that formula feed is just as good as breast milk?
No. This is just showing that their microbiomes are not changing in formula feed versus breastfed. I mean, when the baby latches on, the breast is going to have other things on it. So anything it's not purely breast milk that they're having. There are loads of other things that affect the microbiome. Yeah, but what did cluster was that HIV unexposed babies, which were in red, sort of clustered a bit better, clustered a bit more together than the exposed babies. And if you look at the difference now, again, this is the same kind of graph where this is the variation in bacteria. It's slightly different between the exposed versus unexposed. So obviously whether the mother was HIV positive or not is affecting the composition of the gut microbiome of the child. So again, you can see if you do unsupervised clustering that when you look at the feeding, you can see it's pretty random. So it's not clustering by feeding, but it is clustering by unexposed versus exposed. So then we also again looked to see the correlation with vaccine response. So all the babies get BCG vaccine at birth, and then they get tested then at 6 weeks for these BCG antigens and various other things. So you look at CD4 and CD8. So these are immune cells, and if they're upregulated, then your immune response is up. If they're downregulated, if there are very few of them, your immune system's down. So you measure this by you measure the cells or you measure these cytokines, which again are these immune molecules. And what was found was that there was a difference between the infected versus uninfected, the exposed versus unexposed. So this is again no clustering by feeding, but that's breastfed versus formula-fed. And then the ones that were exposed, so this is low CD4 count, there's basically a reduced immune system. It was very specific for certain kinds of bacteria, so they're clustered separately. And Bacteroides fragilis came up as a very distinct classifier of something that will change your immune system. And in fact, when you go look at the literature, it has been shown to affect the immune system. So it's extremely important bacterium that it seems to be changing in the microbiome, which is changing their response to the BCG vaccine. And this is different between the HIV exposed versus unexposed. So basically, that was just a pilot study, and what it's showing is that there are differences in diversity and abundance of the gut microbiome between those born to HIV infected versus uninfected mothers. An altered gut microbiome is associated with impaired immune responses. So this dysbiosis of the microbiome from having an HIV-infected mother is causing an impaired immune response. So it's not responding to the vaccine like it should, and it's not getting the protection it should from that vaccine. And it's specifically linked to particular bacteria. And so we're now looking at those in other patients, in other infants. So then just to finish off, that's some of the research that we've done specifically. I'll just show some of the research we've done on women. We're also looking at the male genital tract to look at microbiome before and after circumcision, and also related to different behaviors and HIV infection. But because of the Women's Week, I chose to talk about the women research. But there's a lot of, this is now a hot field. Everybody's going into this field because there's so much impact that people are finding now of the microbiome on disease, on vaccine response, on your immunological state. And so the Human Microbiome Project has now been extended. So there are multiple different sites that are being tested. So all over males and females, skin, underneath things, over things, inside things. And these are now being catalogued. And there's a whole lot of new research that's happening in terms of not just what relation to specific diseases, but in order to do that study, people are cataloging all the genes in the human gut microbiome, which will help the study. So you've got a reference genome to look at. People are now saying, okay, let's not just look at the microbiome, let's look at the virome, because now, of course, along with those microbes, there are thousands of viruses in there too. And you won't pick them up from 16S RNA sequencing because they don't happen to have that gene. You have to use a whole new technology to actually now pick out the viromes. And the virome is also showing a significant impact on immunological factors and diseases. So that's a whole new area of study. And And because of this, yes, we need to also think about microbiomes when developing vaccines, because we've just shown that there's it affects the vaccine response. So there's a lot of research now on using fecal transplants. So basically, this is literally feces in a little capsule where they've taken it from a person who has a healthy microbiome, and they give it to somebody who has dysbiosis in their gut microbiome. And that has been shown to change the gut microbiome to become healthy again and cure diseases. So this has been used in diabetes, it's been used in many diseases. I mean, it's still in the kind of the research, the early research stages, but there've been cohorts of patients where it's been shown to be effective as a therapy. So literally just a capsule of feces.
Is that why my dog eats his own food?
Well, maybe you never know. Maybe they're far more advanced than we think we give them credit for. You know, he thinks he's going. He needs to get back that microbiome to his healthy state again.
We used to do that with sheep. You bring sheep from one area to another area where the bacteria is different. You kill one of the sheep from that area and you feed it to the sheep.
Yeah.
So that's been done
for many years, and you know, and this is probably why it works. Yeah. So it's a big new field of study. But what they have found actually in some of the studies is that you give somebody this fecal transplant, and it works actually for a while. But then after a few months or even a couple of years, obviously whatever caused your microbiome to have dysbiosis in the first place comes back and replaces it. So sometimes you have to have these reasonably regularly. And so they're now experimenting with sort of regular therapy of fecal transplantation. So it's a very new field, but it's certainly up and coming, and it has shown definite impacts on several diseases. So I think microbiome research has a lot of potential for health interventions, not only to fix diseases, but really to understand the diseases in the first place. Diabetes, so many people think about the diet and what genes are involved in diabetes, but actually there's a significant impact of your gut microbiome on diabetes and the cause of diabetes, and that's why diet affects it, because diet affects your microbiome. So it's a hot new area of research, and so we're doing a bit of research. Some of the stuff that I've told you about, we're also researching the nasopharyngeal microbiome of babies who come into Groote Hospital to look at the link between their microbiome in the nose and respiratory infections, so pneumonia, and they're finding correlations there as well. So it's new for diagnosis, treatment, and understanding of diseases. Thank you.
How much is really practical now, or is it still all research?
So it's still research, but sequencing is becoming so much cheaper now that actually even for looking at genetic diseases or for cancers, in the States, they're starting to do routine sequencing as a diagnostic. So it's becoming cheaper and more realistic. South Africa's not quite there yet in terms of having the facilities. I mean, it's still, I mean, the BV, for example, you could see, you know, it matched the laboratory results, and for now the laboratory is going to be cheaper. But if you want to sort of fine-tune that intermediate section, that is actually better teased apart by the microbiome than it is by laboratory techniques, because that's an objective view.
You mentioned diabetes, so there are these factors which are normally, you know, well known of diet and heredity. To what extent can you say now that these microbiomes are in fact not simply a derivative of those confounding factors, but are in fact operating completely, you know, as the main causal factor? So
the people who have done research,
subsidiary.
Yeah. So people have done research to prove that by looking at things like, so for the genetics, they'll look at monozygotic twins, so twins that came from, so that are absolutely identical, and they'll do studies on those to check whether it is genetics. They'll put them on the same diet, or they'll do a cohort of patients and they'll treat them so they have exactly the same diet. So they try to make the variables more constant across those so that they can test what really is varying, and there is a distinct difference in there's a distinct correlation between the microbiome and that's not related to disease in particular studies. Obviously, all factors are important, but if you tease apart the individual ones by keeping everything else constant, they have been able to show this.
So you can get to the relative contribution of microbiome as opposed to genetic inheritance.
It's still quite difficult because of the natural variation in the microbiome. Natural variation in the microbiome is still so big that it's still difficult because, you know, if you lick your finger, it's different.
In the case of diabetes again, is there any difference between the onset in childhood and onset in later life?
Yeah, so people have looked at that, and actually your microbiome changes a lot over time. So if you look at the variation in the microbiome, you know, for infants, and then you look at the Human Microbiome Project has looked at different age groups as well, it's very variable, and it tends to kind of get to a plateau. So your microbiome does change over time. So sometimes late onset is actually from the change in your microbiome over time because your genetic factors were there all the whole time. So that sometimes explains late onset versus early onset diabetes in some cases.
I read an article, I think it was in Scientific American, where they were looking at the gut microbiome, and they were saying it could be broadly classified into about 4 different types, and that it's possible maybe to change and do a flush, and then you can just change to a different type. And it's possibly something that we ought to know about ourselves, so that when we go and when we're treating ourselves or someone's treating us, then what flavour of microbiome, what colour of microbiome we happen to have would be important.
Well, that's why, so these fecal transplants will be one of those flavours.
Right.
And so that's why when you have antibiotics, they actually suggest you have these sort of, the yogurts that have got lots of different cultures in them, because you destroy your microbiome. And that then actually leads to many other things. So you can actually cause yourself a disease by destroying your own microbiome by taking too many antibiotics. So that's why there is an impact, because antibiotics is killing your microbiome. Sometimes selectively, sometimes they have some resistance against them. But yeah, so it is possible to change it in many ways. And maybe in the future you'll have your little flavor of fecal transplant that you can pick and say, I'd like that microbiome, please.
You mentioned only one cancer, the colon cancer. Is it the only one correlation that you have?
No, there are lots now coming up. I just picked a few examples, but there are a lot of new cases that they're finding of. I mean, colon because they look, you know, specifically in that area, but there are new papers coming out for other kinds of cancers that are being linked to the microbiome. Because I mean, if you think about it, the microbiome is making chemicals, is making compounds, is making substrates the whole time. And you know, some of those could be carcinogenic, it changes, it causes imbalances in your own system. There's just so many different impacts. If they outweigh our cells by 10 to 1, they're going to have a bigger impact than our own cells, you'd think. Yeah.
Has this work been done on say for example, something like autoimmune diseases like rheumatoid arthritis, for example?
I haven't seen studies on that, but that just could be because I haven't looked. Okay. It's quite possible that it would affect arthritis because the microbiome affects immunity. And
what would the site be that you would take the material from to study that?
So for rheumatoid arthritis, if it's immunological, you'd have to look for the site of, you know, where it's being affected. So maybe, you know, in that case, maybe joints.
Yeah. Okay. What, you take material from the bone?
Yeah, they're taking material from all over the place. So in the joint fluids, maybe. Okay. Yeah. Jeff, did you have a question?
Thank you. That's a wonderful talk, International Women's Day especially. My question is about computational, which fortunately is hidden from us. Just like mostly they're correlation based, are there anything which is too slow that you might compute, or do they all work fine for the computations?
So with 16S it's actually okay. So we use a cluster at UCT, because we have thousands of samples and actually there are millions of reads. So it's just the mapping, it's just a problem of mapping millions of reads to find the reference genome. When you start moving to metagenomics, whole genome sequencing, so now this is not just finding, just sequencing specific gene, you just sequence everything that's in there. That's a much bigger problem because now you're looking at multiple genes. You don't know where those genes are going to be hitting. Some are very similar in some places to some bacteria. So sometimes you have to do de novo assembly, which requires very high memory machines. Another field we're looking at is metaproteomics. So this is, instead of looking at the DNA sequences, you basically just mush up that poo and pull out the proteins, and you run them through a mass spectrometer. And that's a very big computation. We haven't solved that computational problem yet. Because we want to, yes, you can use 16S rRNA sequencing to say, okay, these are the bugs I know are there, so I'll just find those proteins and run my sequences, my mass spec against those. But we want to, so we had a particular patient who had been to every single infectious doctor. They just had a disease. We could not figure it out. It was something in the gut. So we wanted to find out something that you wouldn't find, something novel. So you're not going to find something novel when you're looking at something that's existing. You know, you've already decided what's there and you're searching against that. So you wanted to search against every single fungus, every single pathogen, every single anything. And the computational problem is just too big. One sample of one little mass spec was taking 2 weeks. And we had 100,000 mass specs in a spectra that had to be run. That is still a problem that we haven't solved yet. And we want to solve. Supercomputers, we've gone to the experts of proteomics in the world that cannot solve this problem. Because, and then it's not just a problem of computing, because it's the database size as well. Yeah, but as soon as you change the database size, you change your false discovery rate, you know, the likelihood, everything just goes out. So there are some serious computational problems in metaproteomics. 16S we're still under control for now, but metagenomics also a bit of a problem.
Could it be used for sports people not to use drugs like the big message from this morning? Tennis players taking drugs.
Oh yes. It's yes, because you know what you can do actually is, if you're very clever, is you can work out which bacteria make those drugs. So drugs, a lot of the antibiotics are actually made by a particular bacterium called Streptomyces. Streptomyces make antibiotics, and that's how they got their antibiotics in the first place. Penicillin is made by a bacterium. So if you, I mean, I don't know who anyone's tried it. If you now do an implant of this bacterium and you've got the right conditions, it'll start making this antibiotics. So yes, I think in future they'll be engineering your microbiome to make the things you want to make. You know, and they constantly within them. You know, another project we're doing actually with one of an ex-AIM student is modeling the what's happening in the connection of the interactions between the microbes in the microbiome, because that's also going to, you know, because what's made by one is going to be used by the other, and so you're looking at what's mutually exclusive in the microbiome. So you can use that information to start engineering things like that. In the distant future. In the distant future in science fiction movies. Any other questions?
Very nice talk, thank you.
Thank you.